Hi everyone. Thank you for joining us on this BioSpark LeaderTalk event. So as usual, I would love to start with a general introduction of the BioSpark group. So as well, thank you for many people, especially people from China, who are still enjoying the Chinese New Year festival season. So thank you for joining us today. Many people, I think most of friends here are already familiar with BioSpark. So basically, BioSpark is a nonprofit organization which was registered in Massachusetts. So we are pretty much a group of young professionals keen to learn and grow. So we aspire to build a global network to support each other in pursuit for career success. So our vision basically is to establish a biotech ecosystem with collaborative and supportive network. And we try to promote the leadership and entrepreneurship, and also try to facilitate the communication for different parties, and also try to facilitate the translation of life science innovations as well. So through the past few years, so many great high quality talents across different life science industry and academia have joined BioSpark globally. So we have many memberships across biopharmas, venture capital, academia, consulting company, and law firms, CPA firms, etc. So here is the management team of BioSpark. So BioSpark was co-founded by 4 people. Like we are in different kind of positions. Some people are in the entrepreneur, try to run their own company. Some people like me are still working in industry, and also some people like Jun. He's a professor in NYU. So we are a group of people try to contribute from very diverse perspective to consistently promote the BioSpark network. So within BioSpark, so our mission is basically to use different event channels, try to bring people, talents from different backgrounds, but mutually we try to make sure everyone, no matter what kind of events, which events you join, you are going to get something you are interested out of it. So no matter you are interested in entrepreneurship or growing your career in industry, or you want to establish some scientific or business collaboration, we are confident that within BioSpark you should find your way out of it. Especially for LeaderTalk event, so we are in to learn the leaders from specific areas. Like today, Dr. Peng is very experienced biopharma professional, and also we also invite leaders from academic scholarships, also entrepreneurs and venture capitalists. So they will be able to share their insight in terms of career development or some field expertise, or share some company strategy or their visions for the industry. So, and also, the success of BioSpark cannot be achieved without the support of many very kind sponsorships who continue to support the BioSpark to enable us to focus building the global network of life science. So without further introduction, so we are very delighted to have Dr. Li Peng, the CSO of Palleon Pharma, here as the speaker, and the host for this event will be Dr. Lian Jun Tang, the founding partner of ISWTB. So Lian, please take the stage.
Thank you very much.
Oh, sorry. I'm sorry for this specific event. So I'd love to specifically thank the Insights We Trust Bio Advisory, and also Yiyao Jingliren from China to kindly support to co-host this event together. And later in next week, also I think Yiyao Jingliren will have a relay broadcast of this event online in China as well. So Leon, please take it away.
All right, thank you very much, Devin. Actually, I want to make this really casual. When people call each other doctor, you feel this is too serious. So if you don't mind, Dr. Peng, do you mind I call you Li, and you can call me Leon? So that would be a little bit easier. I want to get your permission.
Sure, Leon.
Okay, thank you very much. As a reminder, this is still a Spring Festival holiday back in China. So whoever dial in from China, you are doing a great job. So that means you really want to learn. You take the precious time off from the time you're supposed to spend with your family, with us, to learn from us. So we'll try our best not disappoint you, make sure your time is worth it. With that being said, I also want to give a little bit shout out to Yiyao Jingliren. This our partner this time. What they would do is they're gonna do exclusive replay of our webinar. I believe 1 week from now, and that time, if you miss the live events today, you can join that one. And also, Lee will join that exclusive replay real time and answer questions. So going to be kind of a semi live events as well. With that being said, without further ado, let me give a quick introduction of Lee, Dr. Li Peng. Obviously, you probably already have read his resume from the flyers. Very accomplished scientist, have a lot to show from our generation, and also is very inspirational. In summary, obviously, she was born in China, as many probably Asian Americans in this country. We went to the States, try to learn the best from the science, and later on found our passion lead us to something probably not like the traditional academic path. And she followed her heart and spent quite a long time in one of the biggest pharma company, MedImmune and AstraZeneca. Afterwards, she made a leap of faith, jump into this biotech company when there's very little to show at that time based on traditional standard. And guess what? A couple of years later, she's leading the company, leading the scientific team of the company, and she's bringing this Nobel Prize winning technology to the next generation of a cancer drug, and many believe it could be arguably best in class for next generation immuno-oncology. So that's a really quick summary for Lee. With that being said, and I will get started. So how are you doing, Lee, today? I see your looks great.
I'm doing well. Thanks, Leon, for the really kind introduction. Thanks for inviting me for this interview. I'm very glad to chat with the friends, with you, and share some of my thoughts.
Yeah, thank you very much. So in the meantime, actually, Devin, can you please stop sharing the screen? Because I think everyone, people
sure.
want to see Lee a little bit more than the screen. The screen looks
nice, I think
but,
that's a good idea for sure.
Yeah, thank you. All right, so I guess everyone just looking at our two pretty smart face. I would not use the word beautiful because we appreciate our intelligence more than our looks, but I think our looks pretty good as well. I'm kidding. So let's get started, Lee. So obviously, two things immediately jump out when people look at the resume. Number one, like I said, you are Asian American or Chinese Americans. Can you just start with a little bit personal? What's the best memory you had when you were in China? Not necessarily in the school, right? Whatever you are happy to share.
Yeah, great question. Yeah, I certainly have lots of fun memories from China. In Boston, we just had the snow. The snow brings me back to my childhood. I grew up in Hunan, where it's uncommon to have snow in the winter. So one year we had about 5 inches of powdery snow. The first time I had seen the world turned into a winter wonderland. And I remember that was a school day. And we had schools in the morning. After 2 hours of classes, my teacher announced that there was no more class for the rest of the day, and the entire class would play snowball fights. And that's my best snowball fighting memory from my high school experience in China.
Wow, that's very good. So you say people always think people like us, we all jump through a lot of loops to get into the best school and college in China. But it turned out the best memory for you is not really studying, the hardest or get the entrance, pass entrance exam to get into good university. It's about the snow fight. So
Yes.
it's just tell you, just like we drug developer, you'd better do something you're truly differentiating, right? This sounds like a pretty differentiated from many other memories. All right, so another question, obviously, why did you choose to do a PhD overseas, especially in the United States?
Yeah, so when I was in college, I fell in love with life science, even though initially I didn't know whether I truly loved biology. What intrigued me the most was the unknown in life science, so much to discover, so many avenues to explore. And so after completing my undergraduate degree in China, I decided to pursue an advanced degree in life science in the US, earning my PhD in biochemistry and molecular biology from the University of California, Davis. I think at the time I made the decision, I would say probably follow the trend, like a lot of college students study life science and decided to come to the US for advanced degree at the time.
Yeah, I mean, obviously, I follow your footsteps. I am a few years junior of you. I mean, that's a really, I would say, one of the best choices many of us made, right? Went to the States. I'm pretty sure you probably get a way more than you expected when you were in China, right? When you made a decision, you want to go abroad. Turned out probably the science is very exciting, but there's a lot more the States had offered to you. That's very good. So obviously your next step also on that time, a little bit, how to say, untraditional. I believe on that time, your professor or probably a lot of your alum would tell you, if you're really passionate about science, you should stay in academia and get a professorship. However, you kind of made your mind really early on after your PhD, you just went to industry. What's going on over there? What was happening in your mind when you made the decision at that moment?
Yes, you're absolutely right. I think the conventional path for graduate students was getting your PhD and do postdoc and apply for professorship. And yes, I went a very different path. So during the last year of my graduate school, I began to consider a career path in biotech. I think it's mainly influenced by my PhD advisor, Dr. Kim Lam, who's an MD PhD oncologist focusing on translating basic scientific research to develop cancer diagnostic and therapeutic applications. He also had his own company while he was my PhD advisor. And I thought it was fascinating that you can translate your own basic research to practical applications. So, and I was very curious about biotech at the time. So I applied for a 6-month internship in the tumor biology department at Genentech, one of the best biotech companies. And this experience really opened a new door for me. And my supervisor at Genentech was very supportive of me and my goals. And she helped me to understand how exciting and rewarding a career in biotech could be. And I really like the innovative, fast-paced biotech culture. So at the end of the 6-month internship at Genentech, I accepted a scientist position at MedImmune, which is the biological division of AstraZeneca, which is another great biotech.
Yes, absolutely. Wow, that's fascinating. I already pick up 2 very important messages. I think some of the junior people, the younger generation could resonate with. Number one, you had a great mentor who pretty much show you the really like new territory. Most of us had never seen before, right? Like you said, traditionally people only look at academia, but you follow this, I would say, scientist entrepreneur, right? In a sense, that he is also a physician scientist. There's a lot of really, I would say, exciting features of your mentor. And also picking up another one, you said when you went to Genentech, your supervisor was a woman. I don't know whether that's something probably, this is, I just making dots, like connecting dots a little bit quickly, because the story is really about how you work with another fascinating women scientist, right, to do something great, which is Caroline Bertozzi. We'll get that later on. So before we jump to that real quick, let's leave it the suspense. Can you describe a little bit your experience in MedImmune AstraZeneca? Like you said, it's a great company. I mean, there's a lot of great drugs from MedImmune, including durvalumab, tremelimumab, right? So I stop there.
Absolutely, yeah. So yes, MedImmune. I started as a junior entry-level scientist at MedImmune, and MedImmune is not just a great company, but it's also a great school for young scientists. And so I've learned so much, not just science, but also how to manage a project from target validation all the way through R&D and working with the cross-functional teams. And I learned how to be effective and not just focus on your own ideas and how to lead a team. So I felt like I was transformed from a very green scientist to a mature professional, biotech professional through the 10 years of a journey at MedImmune. And along the way, I acquired lots of good friends at MedImmune. And of course, I get a lot of support from my supervisors and other senior co-workers while I was junior at MedImmune.
Yeah, fantastic. Sounds like working in the biotech pretty much give you the same training as a postdoc, but you could just get paid much better. I'm just joking. if you choose a good company. Now let's get into I would say, the main chapter of today's, I would say, fireside chat, if you may. Our title already disclosed quite a bit of information, how two women scientists are transforming Nobel Prize winning science to next generation cancer drugs. So before we get that, I'll give people, if you are not familiar why we are saying this, a little bit 30 minutes introduction. So actually Lee, she has been working in Palleon for more than, I think right now it's like 6, 7 years, right?
Yeah, 6.5.
6.5, yeah, I'm pretty close, 6 and 7, so in between. The Palleon actually founded by Caroline Bertozzi, who won Nobel Prize last year because of her contribution to click chemistry. And what she has been doing is use this click chemistry as a tool to understand the glycobiology, especially in inflammation as well as in cancer. And because of that, actually, she founded the Palleon a few years back with a group of very talented entrepreneurs, serial entrepreneurs, and other people also, including Lee. And through the years, obviously, even the science later on won Nobel Prize, there's a lot of challenges and I would say barriers you have to overcome. That's where, Lee really did the heavy lifting to, convert or turn out Nobel Prize winning level science to something pretty promising. I mean, this is pretty much in the coming 20 to 30 minutes. We're gonna dive into this and listen from Lee, her side of the story, how this journey gets started, what's the ups and downs, and how she's like envision the future of this journey can lead us. So with that being said, let's start with something very simple. In the very beginning, obviously, you already said that, spent 10 years MedImmune, you learned a lot, you get an increasing responsibility. A lot of people are going to stay there. That's really comfortable. We all know in big pharma, big biotech, and that time, by the way, MedImmune was pretty good. Why did you give up that such a comfortable, if I may, really good kind of a financially secure position to Palleon based on our conversation at JP Morgan last week. And that time Palleon had nothing, just a pitch deck.
Sure. So give you a little bit more story. So when I joined Palleon 6.5 years ago in 2016, Jim Broderick, Palleon CEO, had already identified this nascent field of glycoimmunology and founded the company Palleon with Carolyn Bertozzi and also Paul Crocker. And also he had raised 47 million in Series A from a syndicate of well-known investors, including a few large pharma companies. However, we didn't announce the Series A until a year later, October 2017. So probably a lot of people didn't know that. And so, yes, Palleon is a little different than most of the startup company because it's not like a spin out from a lab with assets or with targets already. This idea came from the CEO, Jim, first, because he saw the huge potential of this field, and he invited Carolyn Bertozzi, also Paul Crocker, another glycoimmunologist in the field, first discovered Siglec, to form Palleon. So when I was introduced to Jim, and well, I already had read Carolyn's papers and was very inspired by Carolyn's vision, how she sees the potential of this field. And I was deeply intrigued by the novelty of the science and the tremendous potential to translate to a new approach of treating cancer and even autoimmune disorders. Though I wasn't looking for a new job at the time, but I made a very bold decision to relocate my family from Maryland to Massachusetts because it was an opportunity I felt I couldn't pass by. I viewed it as a once in a lifetime opportunity.
I say, wow, that's a really, really interesting story. So I guess you were convinced by the science, just like Jim did himself first. And I'm pretty sure he's a very persuasive man. Actually, I knew him, I met him. we had dinner before. He is very, very good. He is very good at this. But what I heard also from JP Morgan when we chat, it's obviously, the startup is quite different from MedImmune / AstraZeneca, which is, pretty much a two-polar extreme, right? You from something you have tens of billions of dollars of revenue to something you have 10 people, 20 people, right? You have nowhere to make money for another 5 to 10 years. So what was your first month like when you were under Palleon? Did you have any kind of a fond memory from your first month under Palleon?
Yeah, absolutely. So at the time, Palleon was very small. The team of Palleon is 4 people plus me, including me.
So you're employee number 4.
Right, yes. So my first month at Palleon went by very quickly. I was busy reading glycoimmunology papers, meeting with our scientific co-founders, Carolyn Bertozzi and Paul Crockers, and also our SAB members. And lastly, getting to know the small Palleon team of 3 other co-workers. So, and in the first, yes, so it is very different. Like we even didn't have a lab at the time I joined the Palleon. We only had an office in the CIC building in Cambridge. And a lot of times we had team meetings in the lobby of Marriott, which is across the CIC building. Yes.
I see.
So very, very different. Yes.
Yeah.
Compared
Wow.
to a fully integrated company. Yeah.
Yeah. So just curious, how many people you have right now?
Right now we have about 40 people. Yeah.
40.
Yeah. Our philosophy is to build a top performing and lean team. So we were very disciplined about how to use the capital efficiently and trying not to build a too large of a team.
Yeah, that makes a lot of sense. I think this is also philosophy, especially for people dialing from China. which you and I both know very well. That's something they probably should like listen to and learn a little bit. Later on, if the audience get a chance to check how much money Palleon has raised, you'll be shocked. They only have so few people because they raised a lot of money already. With that being said, another question is, obviously you are expert of a glycobiology. Can you use some layman's language to describe to us, let's say if you could in 1 or 2 minutes, what is glycobiology? What is glycan? Why it's so important for cancer cells and even for treating cancer?
Sure, yeah. So glycoimmunology is an emerging field that addresses how glycans, which are the sugar molecules that coat cell surfaces, regulate and direct the body's immune responses. Glycoimmunology is rooted in the field of glycobiology, which is defined as the study of the structure, biosynthesis, and the biology of glycans that are widely found in nature. And we found glycoimmunology just has much opportunity. You can translate such a rich biology to develop drug opportunity, treatment, like novel therapeutic modalities or therapeutics to help patients. I see.
I also, I review your educational center from your website. It's very good. I like, to learn, especially I'm trained as an immunologist, so a little bit easier for me. But for people who are not in immunology, space, when they look at this, I'm pretty sure they can understand as well. Because, Caroline is a chemist anyway. If she can understand, she can describe some complicated immunology ideas. I'm pretty sure anyone who are decently interested in science should be able to understand this. So another question is like, obviously you and I both in IO field for a long time. In case people don't know me, I actually trained in immuno-oncology, and glycobiology has been kind of in kind of academia as a way to target cancer for a long time. But the truth is most of them fail. And can you tell us why it looks like you are the clear front running horse in this kind of a almost 10 plus years race? What did you do like differentiate yourself from other unfortunately setback?
Yeah, absolutely. So I think in the glyco field, often when you come to developing drugs, people treat glycans the same as proteins or DNAs. And that probably is the major cause of not being so successful developing drugs against glycans. With that said, there is approved anti-GD2 therapeutic available. And so we require new framework when handle glycans, because glycans are very, very different from proteins and DNAs. Glycans, the vast structural diversity make it very challenging to characterize and synthesize. Proteins and DNAs are linear structure, glycans are branched structure. And the diversity of glycans is way more than proteins and DNA. And the second, study glycans function is hindered by the lack of tools. And thanks to Caroline, she took the click chemistry to the next level and founded the bioorthogonal chemistry to study glycans in living systems. So with that, she has catalyzed the field, and the glycoimmunology field has really taken off from those wonderful chemistry and tools. I think when Palleon was founded, the field has matured to a degree with a lot of tools and understanding about the function of glycans. In the past, a lot of companies simply treated glycans as a tumor-associated antigen for targeting. So Palleon took a very different approach. We understand the function of cytoglycans. They inhibit immune responses. So we don't treat them as an address to access tumor, but we want to functionally block sialoglycans mediate immune suppression to alleviate the immunosuppression for treating cancer. And with the right mindset, and Jim was able to raise the money and also gather the talents at the Palleon to pursue this mission. So I don't know if I made it clear. So
it's very clear.
The field is mature enough, and also it's converged from chemistry, immunology, and also glycobiology together for this new glycoimmunology. And we took a very different approach at Palleon, developed like Eagle platform and also two other platform technologies to uniquely address the challenges of glyco field.
Yeah, fantastic. Because I mean, actually, there's one podcast, featured Caroline by Freakonomics. I think you and I both listened to that one. It's really interesting, right? It's one economics podcast, one of the best one, arguably. I think she also used a metaphor. I think that's probably layman's description. She described the glycans in like cancer cells look like feel like weeds in a like disarrayed garden. That's why everything was hidden. That's why the immune cells could not recognize it, right? So sounds like she pretty much say the sialidase, which is kind of the enzyme, which is the backbone of your drug assay, looks like a mole, right? They kind of just part of all those grasses, not all of them, but the bad one. And so all those features, like you said, proteins or stuff on the cell surface would be exposed. Pretty much she and you have been working on this like magic molecule level grass mold to make sure the garden is in good shape. So other immune cell and other fancy, smart, intelligent and useful visitor can stop by and put a cancer cell in check. I don't know. That's my understanding from that one. I don't know whether that makes
you
sense.
described it really well. Maybe what I can do differently is to share a story behind the Eagle therapeutic platform. When Palleon was founded, and the company was initially pursue Siglec blockade using antibodies. Siglecs are the immune receptors sensing sialic acid, sialoglycans. And because at the time, investors, Palleon's investors viewed the sialidase approach was too risky. They don't want to layer another modality risk to a novel biology, the risk of novel biology. They preferred conventional antibody approach. That made a lot of sense.
Yeah, so by the way, quite a few companies like Next Cure is using that.
right. Yeah, there are a couple of companies doing Siglec blockade. So when I joined Palleon, and actually after maybe was in the second week, I actually did something quite memorable and bold. I made a case to convince Palleon CEO Jim to change the R&D strategy for developing the sialidase conjugate Eagle platform. And I thought that was quite memorable for me because I was new to the job. I was new to the glyco science, glycoimmunology, and but I from my observation, I felt I should share my thoughts and compare and contrast the Siglec blockade approach with the sialidase approach. Each approach share different pro and cons. The sialidase approach beautifully address the redundancy of this pathway because the Siglecs have so many Siglecs. You block one couple may not be able to overcome the redundancy or being effective to completely inhibit this axis. On the glycan side, if you use antibody or small molecules, those glycans are undruggable. The only way is to use the nature-provided enzyme, which was described beautifully and came up a beautiful idea from Caroline Bertozzi that conjugating an enzyme to a tumor-associated antigen and antibody. Basically, you kind of like park the cytidase on the tumor cell surface and cut off the terminal cytic acid, which is the Achilles heel of the entire biology. Once the terminal cytic acid is gone from all sorts of cytoglycans, the inhibition of cytoglycan is alleviated by such approach. So yeah, so we actually had quite a bit of resistance from both our investors and even our internal team, some of our internal team members, because they felt they signed up for the cyclic blockade approach when they joined the Palleon. Now we're pivoting to the cytidase approach. They were a little concerned. But after a year of grueling work with a very small team, only 1 or 2 people in the first year were working on the Ego project, cytidase project, and the rest of the team, majority of the team, like 92% of the resource were working on the cyclic blockade. But with a very limited resource, we were able to demonstrate engineered human cytidase to replace the bacterial cytidase to address our investors' concern about immunogenicity with the bacterial cytidase. And also we demonstrate preclinical POC data that the cytidase, engineered human cytidase, is very well tolerated and has single robust anti-tumor activity as PD-1, comparable to PD-1, PD-L1 blockade. So that laid the foundation for the Ego therapeutic platform, which is one of the major therapeutic platforms at Palleon today.
Wow, what a beautiful story. This, I have to say, that's a kind of a title of my company called In Science We Trust, right? So in biotech company, probably you should rephrase as in scientist we trust, instead of investor or your business executive we trust, right? I think Jim, I know his science is pretty good. Even he doesn't have a PhD, but he's really, I think, is a model CEO, right? Early on, you should let science speak, right? This is a really beautiful story. Tell people, when you start a biotech, you probably are gonna end up somewhere totally different from where you started. You just have to be open-minded, and also you should always be careful. Don't become, dogmatic, right? I exactly know what you are talking about. And that time, there's quite a few high-profile papers identify different subset, different kind of a cyclic, right? They have a different kind of a blocking antibody, cyclic 15 or something like that, work beautifully. But you have the vision to say that, oh, guess what? There are so many receptors, right? What if they are redundant, right? You block one, other receptor probably going to do the same. But anyway, that's really nice, really beautiful story. That's also a very good transition to next question about the money. Obviously, yes, your investor were not happy. You are pivot to something different. As that's not what we pay you for, right? We pay you to do like cyclic. Now you want to do cytidase, you want to do glycobiology. However, no matter what, your investor stick with you. They did not put their money out. They did not throw in the towel. So actually, that's very important, right? So can you tell us a little bit about the investor? Because I look at them, they're a little bit different from typical biotech investors. Can you expand that a little bit?
Sure. Yeah. So the Series A investors were mainly from large farmers, including a couple of the big pharma investor venture capital firms. And that was designed by Jim Broderick. And he particularly wanted to have such a syndicate to share the long view. And they have the patience to figure out this biology because he knew that this is a new biology. It would require the time to figure out a lot of things because it's new. And there are things you just don't know.
I see.
And I would just say, yeah, our investors, without the investors' support, we wouldn't be able to do all of this.
Yeah.
And yeah, so we were able to navigate through the complexity of the biology and let the data to drive the decision, and being openly, radically open-minded, and let the science to lead to the next step.
Yeah. So for people who are not familiar, so I can give you a quick summary of their syndicate. Just drop a few names. I believe you have a Vertex Venture, right, which is a corporate venture arm from Vertex Biotech.
Oh, no, it's not yet. So
yet.
Leon, Vertex is from Singapore's.
That one also,
right,
like Temasek is that's also very good. Sorry.
Yes, and the rest of the Series A investors are all from farmers, including Pfizer, GSK, Takeda, yes, AbbVie. Yeah, did I forget anyone? No, I think
it's
I covered
it. They don't need to you mention all of them. Actually, I just talked to Takeda venture team's friend. I mean, he involved in this kind of investment, and at least he's familiar with. He's so proud of the invest in you guys. This is actually to leave it the background to our friends or like audience who are not familiar with this. For pharma venture arm, for their major incentive to invest in biotech oftentimes is they want to have the first refusal right of a future innovation that gonna change the whole drug business industry, instead of oftentimes other pure kind of a simple venture fund that try to make profits, right? So the philosophy and their mind is quite different. So when I look at your company's investor syndicate, I was like, wow, this is all blue chip. Those guys are very difficult to convince to put in money, but they are very, very loyal. They're gonna stay with you for a long time until you, and also down the road, right? They are so resourceful, those people. Those company or like your investor, you can, pretty much you already get into the door of doing BD deal with any of those big pharma, right? You already got inside the champion for you. So that's really, really good. That's a very good, kind of a lesson all of us can learn. But obviously not everyone can get such a nice science as you guys get and such a great team. So we jealous of you, but we try to learn as much as we can. Then let's, just to jump out a little bit. Obviously, in addition to the venture, you also branch out through some collaboration, and I was fortunately involved in one of them. Last year, Palleon and my former company, Shanghai Henlius, reached a deal. We licensed the HER2 Cialis assay. We kind of not really in license. We collaborate on this as a development partner. We also kind of going to collaborate with a few other right now undisclosed targets for other indications. Can you share with us what's the thinking behind the from your management? Why did you choose China as the future kind of a overseas market? And also, why did you choose Shanghai Henlius? What specialty, what feature kind of really intrigued you guys to make the decision?
Yeah, sure. Yeah. So, yeah, Henlius is a well-established public company in China with a commercial biosimilar franchise and an emerging novel oncology pipeline. We're very happy about such a collaboration. And this collaboration will benefit from Henlius' established biologic process development and GMP commercial manufacturing capabilities, including more than 800 manufacturing employees, and Henlius' substantial investment in commercial infrastructure as well. And yeah, we feel like Henlius has a very strong track record of providing groundbreaking and cost-effective medicines for patients in China. Palleon believes that our collaboration will benefit from their expertise and capabilities. And also such collaboration would allow Palleon to access the market in China. As a small biotech, we probably wouldn't do trials or sell drugs in China by Palleon itself.
Yeah.
And also, I also want to mention that the first candidate in the BDDO is a bifunctional HER2 cytidase. Henlius already has a commercial anti-HER2 trastuzumab biosimilar. So we believe that the co-development of this HER2 targeted cytidase will benefit from Henlius HER2 knowledge and footprint in the space.
Yeah, absolutely. I really enjoyed the time when we were doing like discussion when I was part of a deal team. It's like I remember we were talking this kind of a marathon dating, right? It's take like more than 3 years since the very beginning.
Right,
So
yeah.
people get ready. If you want to get the best science in the world, it's gonna take a long time to build trust. Actually, I think the time we spend on this before you cut the kind of if you may quote unquote married, the dating times are much longer than the average dating time in the United States, I was saying, before they get married. So we are serious. But now let's just jump out, out of give a little bit like a 10,000 feet view of the field, especially immuno-oncology. So you have been working this for a long time. I've been this for a long time. We heard a lot of kind of chatters and ideas. So from your point of view, what's the next breakthrough in immuno-oncology in general?
Sure. Yeah, sure. Yeah. So it seems that many people are having doubts about the next breakthrough in the immuno-oncology field, since so many ideas and novel IO targets didn't pan out in clinical development. And most of those ideas were founded and tried during the booming time, right after the initial success of PD-1, PD-L1, and CTLA-4 IO therapies. So from my perspective, even though it seems to be the winter season for IO right now, this is when scientists and researchers in academia and industry can come up with breakthrough discoveries and therapeutics, because we can learn so much from what has been tried and build on top of those and come up with new ideas, technologies, and innovations. So I think glycoimmunology is one of those examples, which holds the potential to be the breakthrough in the future.
I say, well said, well said. I mean, I think we're gonna spend a couple of minutes, myself as a former immuno-oncology researchers, this is obviously something quite exciting. That's why we did the deal. So in terms of clinical application, right, obviously when big pharma want to take your assay to the finish line, they somehow have to have some clear path forward to get FDA approval. That's just simply put. Oftentimes when they get approval, especially oncology, they need to go after specific cancer type in specific disease stage, or even like in a specific disease setting. I don't know whether, do you guys have a kind of idea which type of cancers or which type of disease stage, what a kind of a treatment history the target patient population would be? The word would be something you want to go after. Of course, I know this is probably something too early, but if you could comment, share some of your thoughts, that would be great.
Yeah. In the future, we'll publish a lot of our findings regarding like tumor type prioritization and the patient stratification for ECO platform. But what I can share now is we have developed our diagnostic platform called Hydra, which can specifically, precisely measure cytoglycans on tumor surfaces across different tumor types. So this would allow us to not just study this biology, but also be able to prioritize tumor types and patient stratification. And we're learning a lot. We're learning from our clinical development regarding what will be the most responsive cancer types or cancer populations to the desylation mechanism. I
see. Yeah, that sounds very exciting. And also, I remember also, you can correct me, obviously, CL-based assay as a monotherapy already have some effects. But when they combine with PD-1, actually, they kind of elevated the efficacy a lot better. One thing I think right now, a lot of people want to work on is the PD-1 refractory and the resistant patients. Do you have any kind of vision whether you want to go after those patients? Is that good fit for you? If so, why? Or if you're not, why? If you could share some of kind of your scientific thinking behind it.
Sure, yeah. So we use our Hydra technology and profiled 18 tumor types and identified that hypersilation, in other words, high density of cytoglycans on tumor surfaces, is a prevalent signature across a lot of major tumor types. And additionally, we did a correlation study in melanoma cohort in collaboration with MGH and Dr. Boland. In a melanoma cohort, we used the tumor samples pre-PD-1, PD-L1 treatment and profiled the hypersilation signature and did a correlation to the clinical outcome. And we found that melanoma patients with a high hypersilation score measured by Hydra had a worst outcome, clinical outcome, compared to the patients with low cytoglycans. So that suggests the hypersilation might be a resistant, one of the resistant mechanism to IO therapies.
Wow.
Yeah, and additionally, because we have single agent activity in preclinical models, once we demonstrate single monotherapeutic efficacy in clinical development, and that gives us a lot of freedom to do different combinations, not just with approved checkpoint therapies, but other approved targeted therapies or chemo treatments. To help patients, not in those refractory patients, but even at earlier lines.
Yeah, perfect. Now let's just move away from science a little bit, just for a second. What's your role model in biotech industry and why? I mean, you have a really exciting journey.
Yes.
There must be someone give you a lot of kind of a butterfly effects, right? Who is this person and why?
Yeah, I would say I have many role models.
That's what I guess, but you have to pick one.
Right, a different phase of my life. My current role model is Dr. Carolyn Bertozzi.
That's a very easy. I loved her as well. After I listened to her, I already kind of loved her. After listening to her like a pre-economics interviews, man, this lady is special.
Yeah,
right?
She's a inspirational and a visionary scientist and leader. She's also a wonderful person, very energetic and charismatic.
Yeah, wow. I'm looking forward to meet her, actually. She's going to give a keynote speech and IO 360 next month. I'll try to, grab a selfie with her. All right, with that, actually, there's a lot of questions from the audience. I'm going to ask them, bring them to you, Lee, if you don't mind. The first question actually from Li Xiu Li, I believe. So the question is, what do you think is the most critical, important things in a startup environment?
Well, great question. So to me, it's make the right decision, use your capital efficiently, and also have a team who's really good at execution as well.
I see. That's a very good answer, especially given the current quote unquote capital winter, right? So you really have to have a discipline, and also you have to have a quality execution. Like, remember, you keep saying this word, this phrase when we were chatting, quality decision and quality execution. I think you hit like the nail by the head. Another question, I think this is from sorry, I butchered her name, from Dr. Tadin Jamal. And this question is a little bit technical. How do you manage anti-glycan antibodies with emphasis to their low affinity for antigen recognition? I'm not sure, yeah, this is a question.
Yes. Can you repeat the
I'm
question?
going to repeat again. How do you manage anti-glycan antibodies with the emphasis to their low affinity for antigen recognition? I know this is a little bit unclear. I guess what he wants is like, we know the anti-glycan. I think that this member misunderstood. This is not an antibody, right? This enzyme. I probably can answer that for you, and you can add. So I think they still thought this is kind of something similar to Cyclic 15 or something blocking antibody. You are blocking glycan. No, we are not blocking glycan here. We are chop off glycan. So
Right.
yeah, that's what
we use enzyme
yes.
to cut off the static acid from the cytoglycans. Yeah.
Perfect. Okay, so another question from Hou Yafei. What is the comments on the potential application of protein glycosylation in drug delivery? For example, deliver monosylated glycoprotein into lysosome through M6P receptor. That's very specific. No
Very technical. Yeah, absolutely. Yeah. So I think there could be a lot of applications using glycan-mediated internalization. In fact, some of the enzyme therapies developed by Genzymes are using the mannose receptor to get it internalized to get to the right place.
Perfect. Okay, we're gonna take 2 more questions because we're running short of time. We try to make sure everyone still can go to sleep early or start your day. This is one I actually quite relevant. I think this is from a student. What suggestion? Oh, by the way, this is from Chen Anran. Chen Anran. What suggestion would you give to the graduating PhD students who are interested in industry? Thank you.
Well, I would say do good science, publish good papers, and submitting your resumes to the jobs you want to apply. I think somehow even at Palleon and some other companies in the Boston area, we felt it's so hard to recruit talents. So, well, if you're really interested into a career in industry, please try submit as many resumes as you can and to those jobs you're interested in.
Yeah, I think Anzan, the coded languages, send your resume to Lee. And she'll take a look. If I'm the translator for you, that's our coded message typically. So this is the last question. This is from David Chao. How did you convince the team and investor to do a scientific pivot only in 2 weeks into a new job?
Right. Yeah. So that's people described me. I was bold at the time. Yeah. So first I was hesitating as well. So I wasn't sure whether it can be effective or not. But I truly believe have the strategy, R&D strategy of including the solid-based approach. It's important for the success of the company. So I started with a mild approach just to ask question with to the CEO to understand what are the concerns not doing the solid-based approach. And with such a dialogue conversation, I was able to share my thoughts and reasonings behind doing the solid-based approach. And with that, we came up with a business case, and I also presented the case at the first Palleon's board meeting. And the board decided to give us a chance. And with a managed risk of, with a proposal with a managed risk, only use less than 10% of a resource to try this strategy and to give company the huge potential in the future. I think that's the major point to make to convince everybody in the board to go along with us.
Yeah, that's such a beautiful lesson, right? So, science, sometimes we know very little, right? To borrow one of a kind of a statesman's saying, I forgot the name, there's a known known, there's known unknown, there's an unknown unknown. We, all of us, especially as scientists, we need to be humble, right? We just have to appreciate and have a good estimate the known unknown, and we have to really have a lot of how to say humbleness in front of the unknown, because guess what? There's so much we don't know, and we always have to put aside a little bit resources for that. And oftentimes that's where you actually make the biggest break. With that being said, we want to conclude today's, I would say, webinar, interview, you name it. And I want to finish with something a little bit personal, a little bit inspirational, up to you, Peng. So the last question to you is, what's your favorite book? And what's a favorite quote from the book?
Yeah, sure. So there are so many good books I like. It's hard to pick a favorite one, but I'd recommend The Principles by Ray Dalio. So he shared the principles that have helped him to succeed in life and work, in the hope of helping others to uncover your own principles. So I was deeply intrigued by his message and the principles of being radically open-minded and learn to make decisions effectively.
Yeah. By the way, both Lee and I are Libby fans. I'm a small fan. Probably she's a big fan of Ray Dalio. He's a great man. And if you don't know him, he's a founder and a long-term chief investment officer of a Bridgewater Associate. It's a little bit different. you like someone from finance. But with that being said, thank you very much. I hope the audience enjoyed it. I definitely enjoyed it, and wish everyone from China a very happy New Year. And we will see you again next time with the BioSpark LeaderTalk Series. Thank you, Lee, for take time speaking with us, and I hope you enjoyed this interview or chat as well. Thank you very much.
Thanks, Liang. Thanks for having me.
Bye.